And welcome again. So now today we will be discussing about the acute inflammatory response. I'm Dr. Ranjit Sai, an infectious disease expert as well as clinical research from the Harvard Medical School. Now we will be talking about this acute inflammatory response. We are on page around in chapter 4 where of Kaplan Microbiology, where we are discussing about this acute inflammatory response. Talking about this acute inflammatory response, before going into detail, let me give you an overview to understand the details of these events. So suppose there is a tissue injury, tissue is damaged, or there is a breach in the tissue due to this bacterial invasion, and there is a bacterial invasion. What will happen? There will the tissue damage. This bacteria has been engulfed by this antigen presenting cell, say macrophage or dendritic cell. Then they will release the chemokines, chemical mediators, by which these signals will transfer to your nearby blood vessels. So what happened? There are the tissue has been breached, there is damage, bacteria have entered inside that, and now that bacteria has actually evade your first line defense. Now cellular mechanism has come and play into the role. So there is the innate immune system, cellular response, and the complement activation of innate immune response that you have to understand, not of the adaptive. Now initially there will be the complement activation as well, because alternative pathway and this mannose binding lectin pathway doesn't require antibody to activate this complement. So initially there will be this bacteria will be engulfed by the macrophage, by the dendritic cell, and the complement pathway will be activated by the alternative and that mannose binding lectin pathway, because they require only the microbial surface to get activated. So once these things get activated, now the macrophage engulf macrophage. When the macrophage engulf this bacteria, what will happen? This will release this ingested the macrophage which has ingested microbes under it will release this interleukin 1 and TNF alpha. So this interleukin 1 and TNF alpha will be released. The release of this leukotrienes, the release of the prostaglandins, the byproduct of your complement pathway, that is the C5a, that will act on the mast cell, and there will be release of the histamine, prostaglandin, and leukotrienes. So what is going on? There is a bacteria that has invaded your tissue, that has come into the tissue. Now the macrophage dendritic cell has going and fight to that bacteria, and they are releasing this interleukin 1, interleukin 6, and TNF alpha, prostaglandin, leukotrienes, the complement pathway also releasing this what we call C5a, that is acting on the mast cell and releasing histamine, prostaglandin, and leukotrienes. What is happening when these cytokines or chemical mediators are released? This will act on the local blood vessels, so nearby blood vessel. In the post venule capillaries, what will happen? Due to this prostaglandin, due to the histamine, this will goes under vasodilation. Once there is a vasodilation, the blood flow will be slow. So once there will be a slow blood flow, now what happened? This neutrophil, which is the first marker to come for the inflammation, to fight against the inflammation, what will happen? They will come. How they will come? There is a step by step. What will happen? Initially, this prostaglandin or say histamine will dilate the blood vessel. The neutrophil which are flowing in the center will now comes to the periphery. Once they reach to the periphery, they need from the blood vessel to reach to the tissue damage side or where the bacteria is present. So what will happen? Initially there is a vasodilation, this neutrophil has come to the periphery. Now due to the release of this interleukin 1 and TNF alpha, there will be a certain changes in the neutrophil as well as the endothelial cell both. So there is a changes in the neutrophil as well, there is a changes in the endothelial cell as well. Initially they all are normal, there is nothing on the neutrophil cell, there is nothing on the your endothelial cell. So this bacteria doesn't get this neutrophil phagocytic cell doesn't get trapped, and they doesn't need to come out or goes to the periphery and attached. So initially the blood flow should be normal, should be laminar, but when there are the tissue damage, when the chemical signals has been sent to your blood vessel, now they are what will do normal this interleukin 1 and TNF alpha, then there will be the configuration change on this endothelial cell as well as to your this neutrophil. So there are the changes in the endothelial cell as well as to the neutrophil as well. So what is happening? This interleukin 1 and TNF alpha, they are causing upregulation. There is the formation, there is a protrusion of this selectin called this is the endothelium. So E-selectin, E4 endothelium, selectin and P-selectin. This will be from the Weber pellets bodies which released from the P-selectin. So this E-selectin and P-selectin will be normally formed on the endothelial cell. You just remember about the E-selectin because it is E4 endothelial, and there will be some of the selectin will be formed or say protrude or upregulated in your neutrophil as well, and that is called this is leukocyte, so L-selectin. So you have to understand L. They are actually mucopolysaccharide that has been upregulated on the neutrophil surfaces. So they can call as a L-selectin or also as the mucin cam. So we can discuss about this. You can see over here, initially this chemokine or chemotherapy factor, there is a mucin-like cam. So this is upregulated in your neutrophil. So this mucin-like cam will be regulated on the neutrophil, and the E-selectin will be regulated on your endothelial cell. So this is L-selectin, this is E-selectin. Now what happened? This both will try to, initially there was a vasodilation, this has come to the periphery, then now there is upregulation of this E-selectin and mucin-like cam, that is called L-selectin, and they will try to bind to each other. But due to the force of the blood that is coming from the backward will actually force them, and they will move. They will be attached and detached, and there will be a rolling. So they will be rolling. They will attach to this, again detach, then again attach, then again detach, and in this way they will continue the rolling, unless they will be tightly bind. For tightly bind, what will happen? The chemokines that has been released. Normally you can see the chemokines interleukin 8, which is over here, they will actually go and bind to this receptor on your neutrophil. So in neutrophil, this chemokine, normally interleukin 8, then is in the C5a complement byproduct or leukotrienes B4. So this all will go and bind to these places in the neutrophil. These are the chemokines, they can easily diffuse, and there will be the configurational change of this your neutrophil, and there will be the formation of this integrin in the neutrophil. So neutrophil now will, initially this integrin was not there. At present, when there was no inflammation, even the mucin-like cam was not there. It was upregulated, and E-selectin was not there. But due to the damages, due to the chemical signal they have sent to the local blood vessel, now they have formed endothelial cell have formed E-selectin, and mucin-like cam is formed under phagocytic cell or neutrophil cell. After that, they will attach, detach. So this will continue. This will be continuing. So for tight binding, there will be the chemokines will come to the phagocytic cell, and they will this chemokines interleukin 8, then the C5a, or you can say also the leukotriene B4. This will cause the configurational change in your lymphocyte, and there will be the development of this integrin. So integrin will be developed on the neutrophil, and due to the release of this interleukin 1 and TNF alpha from the macrophages, they will also cause the upregulation of the ICAM, that is the immunoglobulin superfamily chemical addition molecule on the endothelial cell. So initially this was the E-selectin on the endothelial cell and mucin-like cam on the neutrophil. Now there is a chemokines that has upregulated this integrins, and there is also upregulation of the IG cam on the endothelial cell. So what is happening now? This integrin and ICAM will bind tightly. Once they can bind tightly, now this neutrophil has been trapped to the endothelial cell, and then they will actually bypass. They will actually, you can see this is the rolling. Then there will be the activation. It will be the arrest by this integrin IG superfamily cam, and then there will be a transepithelial migration. So they will migrate. Once they will migrate, what will happen? Once they comes out, after coming out, they will go by the chemotaxis, because there is a tissue damage. So there is the more chemical present over there, and due to the chemical gradient, here is more, here is little less, here is little less, here is little less, here is little less. So there is less, there is more, there is little more, there is more, and at the site where there is actual bacterial infection, there is more cytokine, more tissue damage, more chemical signal. So through that chemical signal, they are detecting, they are smelling that chemical signal, they are recognizing the chemical signal, and then move to the same pathway towards the chemical signal and reach to the bacterial infection site, and then they will phagocyte that bacteria by the phagocytosis, where they will kills that bacteria under the phagosome, then libosome, and after releasing the neutrophil itself, we release all the granule content from inside into the environment, and then that neutrophil die and become pus. And they call the macrophage to clear all the infection, to repair all the infection that is present over here. All the inflammation will be shut down by the macrophages again. So you have to understand how this tissue damage occur, what are the chemicals they release, how this chemical signal goes to their local blood vessel, how then they dilute that blood vessel, then the phagocytic cell get marginate, then the release from the macrophages, that interleukin 1, TNF-alpha, prostaglandin, leukotrienes, these chemical signals will activate them, they will force them, they will irritate them, they will stimulate them and form this selectins on their surfaces. They will form the selectin on the endothelium, that is E-selectin. They will form the selectin on the phagocytic cell, that is L-selectin, or mucin-like chemical addition molecule that CAMP. Then it is just start rolling. In rolling, that will not be completed. So we know then they will start this rolling. They will start this rolling because one is attaching, detaching, then there will be a more chemokines will come into the your phagocytic cell, this chemokine 8 or say complement 5A or leukotriene B4. There will goes the conformational changes on your phagocytic cell, and there will be the outgrowth of this integrin. Once integrin will be outgrowth, there will also the this outgrowth of this I superfamily CAMP, that IG CAMP on the endothelial cell, this endothelial cell by this interleukin 1 and TNF-alpha. Then what will happen? This will get arrested. Once is activated, so from integrin, then arrested due to ICAM and integrin addition molecule. Once they are arrested, now they will be transepithelial migration. They are through the migration, they will migrate out, and once they are migrated out, they will, by the chemical chemotaxis method, by sensing the chemical gradient, they will reach slowly towards the infection site, and in 5 to 6 hours they will reach to the site and clear. They will attack the bacteria and cause the phagocytosis. So hope you are understanding this all the procedure. Initially the neutrophil comes in 5 to 6 hours, in 6 hours, and once they reach over there, they will also send the signal by the neutrophil now, and they will call this monocyte, this which is become macrophage, then the eosinophil. This will come outside after neutrophil has reached over there. So once neutrophil reach there, the neutrophil will send signal towards your local blood vessel, where this monocyte, eosinophil or basophils will come outside towards the inflammatory cell, and then they will prolong they will try to fight the infection. That is the process. That is the process of your acute inflammation. Now let's go through the text. What is the text is telling us? So acute inflammatory response, antigens are normally introduced into the body across the mucosa or the epithelial surfaces. Okay, the acute inflammatory response actually is often first response to the invasion and represent a response of an innate immune system. So we know that this is the acute response, this is the first response, and this is the response of your innate immune response. The first step is the acute inflammatory response, is the activation of the vascular endothelial, is the breach of the epithelial barrier. So once there is the breach in the epithelial barrier, so inflammatory response will occur by activation of the vascular endothelium. Cytokines and other inflammatory mediators released in the area result of the tissue damage induce expression of the selectin. So there is the tissue damages, and due to the tissue damages there is a release of the cytokines, and these cytokines cause the expression of the selectin molecules on the endothelial cell as well as in the phagocytic cell. Neutrophils are the first cells to bind to the inflamed endothelium and extravasate it into the tissue. So neutrophil will be the first to get captured and then thrown out toward the inflammatory site. So neutrophils are the first to bind to the inflamed endothelium and extravasate into the tissue, peaking within 6 hours. So within 6 hours, they should reach to the inflammatory site. Monocyte, macrophage, and even eosinophil may be arrive 5 to 6 hours later in response to the neutrophil release mediator. So initially there is a tissue damage that has called the neutrophil. Now when neutrophil reach over there, the neutrophil will release the mediator, and that will call the this macrophages, monocyte, macrophage and eosinophil again in 5 to 6 hours. So in our 12 hours, your all macrophages, monocyte, eosinophil will be at your inflammatory site. The extravasation of the phagocytes into the area require 4 sequential overlapping steps. That is, one is rolling, that is phagocyte attached loosely to the endothelial by the low affinity selected carbohydrate interaction, E-selectin molecule on the endothelium, that you have to understand. So there need to be rolling. How rolling? E-selectin molecule on the endothelium and mucin-like addition molecules on the phagocyte, that is membrane, bind to the cell briefly, but the force of the blood will flow into the area, cause cell to detach and reattach repeatedly. There will be the rolling of the endothelial surface until strong binding is elicited, and then the strong binding is due to your this strong binding. Why strong binding will occur? Because due to the integrin and the this ICAM addition, that is the strong binding. So how they will found? Initially they need to attract, they need to activate the chemokines released in the area during inflammation, such as interleukin 8, complement split product C5a and N-formyl peptides of the bacterial surfaces produced by the bacteria bind to the receptor on the phagocytic surface and trigger the G-mediated activating signal. This signal will cause the conformational change in the integrin molecules in the phagocytic membrane that increase the affinity for immunoglobulin superfamily addition molecule of the endothelium. So the initial thing is that once there is a rolling, attaching and detaching, they need to be activated, and for activation there is the signal like interleukin 8, C5a and N-formyl peptides of the bacteria. They cause the G protein mediated activating signal, and that cause the conformational changes in the phagocytic membrane, leads to the formation of the integrin on their membrane, which bind with the endothelial superfamily complement addition molecule. Then what will happen? They will be arrested. This interaction between the integrins and the IgA superfamily addition molecule, ICAMs, might cause the mediates the tight binding. There will be a tight binding and phagocyte into the cell. This integrin and ICAM interaction also mediate the tight binding of phagocytes and their movement through the extracellular matrix. So now they are arrested, and now the phagocytic cell extends pseudophoria through the basal wall and extravasated into tissue, which you can see in this pictures. You can see this was initially rolling, then there was the activation, conformational changes, then they were arrested due to this integrin IgA superfamily, and then finally they migrate toward the transendothelial migration. Now one thing you have to remember, that is this is leukocyte addition deficiency, that is called the LAD, and if this integrin, if integrin is unable to form, then what will happen? They will develop this leukocyte addition deficiency. Means once this integrin is not there, what will happen? This leukocyte addition deficiency will be developed. So you have to understand in such a way, like this integrin needs for the tight binding, but if that integrin is absent, this disease is known as the leukocyte addition deficiency. Actually, one of the component of this beta 2 chain of the integrin molecule is absent, and this is also called CD18, and since this is absent, there will be no tight binding, so they cannot, this epithelial cell, this phagocytic cell will not come outside into the tissue, they cannot be reached to the inflammatory site, they cannot come out from the blood vessel, and that is called leukocyte addition deficiency. In such case, what will happen? There will be the inflammation, but this phagocytic cell will not reach there. So this phagocytic cell will be actually high in number in the blood vessel, but they are not able to come out from the blood vessel and reach to the inflammatory site. So on inflammatory site, there is no inflammation, there is no pus cell, there is no abscess, but inside the blood vessel there will be high number of neutrophils. Okay.